Okay, so right now our cells are enjoying a dreamy slumber at minus 80 degrees Fahrenheit in the freezer. We're going to wake them up in just a moment, and before long they will be frolicking happily on the surface of whatever plate or dish we put them into. But first we need to remove them from the environment they were in while they were in the freezer. So when we freeze the cells, we need to make sure that ice crystals don't form, because that would destroy the cells. And so when we froze them, we used something called DMSO, which prevents the ice crystals from forming. Unfortunately, DMSO is toxic, not so toxic that it kills the cells. In fact, it's necessary to keep the cells from dying while they're being frozen. But once we warm them back up, it would kill the cells before long if we didn't remove it. So we need to get rid of the current medium that they're in. It's actually, they're directly in the fetal bovine serum, but it has, I think, 10% DMSO, and we need to get rid of that old serum containing DMSO. So I'm going to explain how it works on the whiteboard first. So right now, the cells are in a little microcentrifuge tube, and this microcentrifuge tube has maybe some clumps of cells at the bottom, and the medium itself is FBS plus DMSO. That's the thing that would kill the cells once we get them out. And here's the little clump of cells at the bottom. Okay, so what I'm going to do is, whenever I'm working with cells and I'm pipetting them up and down, I can't use the micropipette or any of the micropipette tips. The reason is that the aperture of the tip is so tiny that as the liquid passes in and out of it, the shear forces are extremely high. Those shear forces would rip apart the cell. Think of, like, if you were at a water park, you might enjoy going down a little tunnel that's like a water slide because it's really big. But how would you like it if you got, flushed down the bathtub drain? Well, it's too small. You would get ripped apart. That's not really a perfect analogy because, of course, the cells are smaller than the aperture of the micropipette tip. But the basic idea is the smaller the opening, the higher the shear forces. You want to be gentle on the cells. So we're only going to use the larger serological pipettes. We'll use the 1 or 2 mil serological pipettes to move the cells. So the first thing you want to do is you're going to have a conical tube, and this conical tube is going to have plating medium in it. Plating medium. You're going to withdraw some of the plating medium in your serological pipette, just 1 or 2 mils. You're going to go over here to the little microcentrifuge tube containing your cells, and once they're thawed, you're going to slowly draw up the FBS, and you'll see the clump of cells, and you're actually going to try to draw up the cells themselves. And you're going to try to just aspirate them up through the little opening of your serological pipette. So I'll show that here. Here's the little opening of your serological pipette. And the little clump of cells, you want it to go in and out, to gently break it up so that it's resuspended in the medium. Be gentle, but you can go in and out at a reasonable pace. Once you're pretty confident that the cells are up into your serological pipette, we're going to go ahead and gently release them into your conical tube. So at this point now, your conical tube has plating medium plus FBS plus DMSO plus your cells that are all resuspended. So the cells are resuspended throughout, but there's a problem. This medium has DMSO. It will kill the cells if you leave them in there too long. How are we going to get the cells out of this DMSO containing medium? Well, think about it this way. Which is easier? Let's say you have a little pet fish in a goldfish. You have a goldfish and he's in a goldfish bowl and you realize there's something toxic in the water. Which do you think is easier? To take the poison out of the water, or to just take the fish out of the water and put it in new water? Well, obviously it's easier to take the fish out of the water than to take the poison out of the water. The problem is, how do we take all of these cells out? Like, they're so microscopically tiny. Well, that's where the step density gradient method comes in. Do you remember maybe back in elementary school, you might have done an experiment where they were teaching you how different liquids have different densities, and maybe you got maple syrup, and you got water, and you got oil, and you put them into a glass, and you saw how they formed nice layers, and then you got like a rock and a grape and an ice cube and a cork, I don't know, something like that, and they all sat at different levels of the layers. Well, it was preparing you for this moment today. What we're going to do is we're going to get another little vial, and this vial is going to just have new FBS in it. So a new microcentrifuge tube. This is just pure uncontaminated FBS. I'm going to take my, my serological pipette. I'm going to withdraw this FBS. I need to put it down at the bottom here so it forms a separate layer. And then what I'm going to do is I'm going to use centrifugal force to pull the cells down into the nice new FBS. Now, this isn't quite like maple syrup and water and oil. Those separate pretty easily. The FBS is more dense than the plating medium, but not by that much. So we're going to have to be very careful. I'm going to withdraw my FBS. Then I'm going to put my serological pipette very carefully down in here. I'm going to lower it in until it's almost all the way at the bottom. And this is where it gets a little complicated because you're looking through 3 layers of glass now. You're looking through the sash of the biosafety cabinet. You're looking through the wall of your conical tube. And you're looking through this serological pipette that has some new FBS in it. Thankfully, the FBS is a darker color and you'll be able to see it. So you'll be able to see like your 1 mil of FBS in here inside the serological pipette, inside the tube, inside the biosafety cabinet. And what I'm going to do is I'm going to very, very, very carefully expel this FBS until it's sitting in a nice layer at the bottom of my 15 mil conical tube. The crucial thing as you do this is not to push it out so much that an air bubble comes out at the end. It's okay to leave a little bit of FBS left in your serological pipette, because if you have a bubble that comes out, the bubble will go and it'll disturb the layer, it'll be all mixed up, you'll have to start again. So I'm going to very carefully expel it, and then I have a nice layer of FBS at the bottom. I'm then going to gently centrifuge the whole thing. All the cells will get pulled down to the bottom to be in a little clump at the bottom. And then I can aspirate this old medium containing plating medium, old FBS, DMSO, and no longer the cells because the cells have moved down to this new layer inside the new FBS. Cells plus new FBS. That was taken from over here. This is the new FBS. Okay? Then once I've got rid of the old plating medium that had the DMSO, I can then resuspend the whole thing once again in some new plating medium. So that's the overall approach we're going to use, and now I'll demonstrate it. All right, so we're ready to begin thawing our cells. So I'm going to get my cells out of here. I open up the -80. I reach in here. If I were working for a long time, I might need the cold gloves, but this will just be a short while. So I close the freezer carefully. Now I'm going to go and warm up my cells along with everything else. So I pick up the little float. I set this carefully in here. I also have a thing of new FBS, as I mentioned on the whiteboard. And I have 2 different vials of my plating medium. So I'm going to let those all warm up. And once they're warmed up, we'll keep going. Okay, let's see. Oh, that's this one now. Okay, we'll pretend it's warmed up and I can just keep going. Okay, everything's warmed up, so I'm going to get it from the water bath. Of course, the water in the water bath is especially likely to be contaminated because it's warm. Who knows what could be growing in there? So I'll want to really carefully spray down everything that I'm taking in from there. I raise the sash to its proper position. For the sake of the video, I'm actually going to raise it higher since I'm just demonstrating. And now I'm ready to begin. So these little vials are handy because they can turn every which way. I'm going to have my my cells, my new FBS, and these might be safe standing there, might be not be. Maybe I should go and get an extra one. Let's see. Again, anything going in I want to very carefully spray down. Okay, now I can have these standing here properly. All right, so I'm ready to begin my step density gradient method. So one thing you'll notice is I very carefully labeled all of my tubes, even before I put anything inside them. That's very important. Not just because you don't want to label them once they're covered in ethanol, but also even before you begin outside of the biosafety cabinet, before you add anything to anything, you want everything to be properly labeled. Okay, so I'm going to open one of these. This is a 2 ml serological pipette. It has a small tip, but it's still going to be much bigger than a micropipette tip. So the way you open these, they don't have the nice two-sided paper and plastic packaging, so I just pull this down and kind of ram it through the end. And I'm going to take my pipette controller, press it firmly on, carefully remove this, set it over to the side. And then I have 2 of these. One of them I've labeled that my cells are actually going to go into. The other one is the new plating medium to add at the end. So once again, I think I'm going to just set this down carefully. I'm going to carefully draw up 1 or 2 mils. Set it down here. Carefully replace the cap. I don't need to screw it on. As long as I'm careful not to spill it over, nothing's going to happen to it as long as the cap is on. And then I'm going to pick up my vial of cells. Now here's where it's a little hard to see what's going on, so feel free to adjust the camera. And let me just turn off this alarm before I keep going. Okay, so it's very hard to see, but if you look right down at the tip, there's a little glob of cells down at the bottom that I can see. It's translucent, it's hard to make out. The only reason I can even see that it's there is that it just looks a little bit more clear, actually, or a little bit foggy, not quite as clear and golden compared to the DMSO-containing FBS that's above it. It's okay if you can't see it, but here's what I'm going to do. I'm going to carefully open the lid, and I'm going to need to pipette up and down to get those cells resuspended. So I'm going to carefully insert the tip, being careful not to touch the tip to the wall, and I'm going to draw up and then down. And as I move down, I might actually move my serological pipette up to help decrease the volume inside the microcentrifuge tube so I don't overflow. But basically, I'm going to move it in as I go down so I can get the cells, and then I move it back up as I go up, in and out, in and out. You know, not too slowly, not too quickly, and being careful not to overflow. And the main thing is I want to make sure I have up that clump of cells that's at the bottom. I don't see the clump up there, so I'm pretty sure my cells are resuspended in here. So just to be extra cautious, one more time, I'm going to push it in. I really don't see anything down there anymore. I think they're in. I'm going to pull them back up. And I'm not going to use this anymore, but it's just good practice to close the cap. And I'm going to return it to the last little bit of plating medium that was left in here. So I just carefully expel those cells down into there. Can you see things okay? How do you think it's going?
I think so, yeah.
Okay. All right, so just for review, this is now a mixture of 4 things. It was plating medium, but I put in the cells and the FBS, and they contained the DMSO. So those cells are there, but they're thinking, this DMSO, I don't like it, I'm not happy. So what I need to do now is put in my new FBS. So I'm going to set these down, get out my new FBS, which I've carefully labeled. And I'm going to very carefully withdraw it. Now, you might wonder, when do I need to change my tip? When do I not need to? Here, since everything is going to be in contact with everything else anyway, I don't need a new serological pipette because it's not like I'm reusing any of these things. So I'm going to take in the FBS, and you'll notice that it's almost impossible for there to avoid being a little bubbles at the top and a little bubble at the bottom. Neither of those is bad. It's okay to push out a bubble at the beginning. I just don't want to push out air at the end. So I'm going to carefully lift the cap off of my cells, bring them back. Now I'm going to carefully insert this all the way down. And usually what I do is I turn it so that the label isn't blocking things. And now it's hard to see, but hopefully you can see this serological pipette inside of the plating medium. Now, it's helpful, these bubbles at the top are actually helpful because they show you where the top of your plating medium is. As I slightly go up or down, you can see those bubbles move. So I'm going to stick them in here, and oftentimes I try to hold it like this so that I can see through. And you can see right now the top of that FBS is around the 12 mark on my conical tube. So I'm going to just set it at the bottom. Here I'm actually going to just rest it at the bottom of the tube, and I'm going to slowly expel this FBS. And at the beginning you won't even see anything moving at all. Okay, it's moving. It's at now 11, 10, 9, 8, 7, 6. I'm at the 5. Now I'm actually below the level of the plating medium. 4, 3, 2, and I'm not going to go all the way down because I don't want to risk pushing out bubbles. So once it gets down to about where the 1 would be, I'm going to carefully remove it. Okay, I'm done with this now. And if you look at this, you can see just a beautiful situation. It looks like some sort of, I don't know, tropical smoothie that's strawberry on top and mango underneath. And I have nice layers. I don't want to agitate this or shake it in any way because it wouldn't separate out again if I did so. But the old plating medium with DMSO containing FBS and cells is up here. That's the pink layer. And then brand new FBS underneath. And I'm going to go and centrifuge it now so that the cells will move down to that bottom layer. So we'll just go over this way. All right, so here's our centrifuge. We actually have 3 different centrifuges. There's the micro centrifuge and the mini centrifuge in the BME lab, but the big centrifuge we keep here in the fluids lab. So I reach back behind here to turn it on. The switch is actually down here. You have to just kind of feel for it. I turn it on. It takes a moment to start up. Now, this lid is notoriously hard to open. Basically, what you have to do is you kind of have to already be pulling up on it when you press the open button. It's just tricky. I can almost never get it the first time, but I kind of apply some gentle pressure. I push open. I can't believe it. It actually worked the first time. Now, inside here, we always keep some extra conical tubes for balancing. I can take out the 50 ml tube. I don't need that. I have one with water on this side. I place this carefully onto the other side. I carefully lower the lid, and I am going to set it at 800 RPM. The time will be 5 minutes, and I just push start. Okay, the centrifuging is done, so I'll once again open it up, and oh, see, it kind of wrestles with you. It says, no, I'm not going to release your samples. Please open. Please, please. Oh, there we go. If you ask really nicely, it'll give you your sample back and then try to slam the lid on you. All right, so there we go. I'm still separated, and I can see a nice clump of cells at the bottom. It's not quite as distinct layers as before, but that's okay. I have a golden layer at the bottom, and the cells are down at the very bottom. So I'm going to be sure to remember to turn this off before I leave the room. Oh, just one thing to note, when you have on gloves, we assume the gloves are contaminated, and so I try to avoid touching any handles or other just surfaces that would be touched by the general public. For example, this door here, if I needed to, I propped it open, but if I needed to open or close it, I try to just do it with my elbow, or another technique is you can just take the edge of your lab coat to open the doors. That just makes sure that you're not contaminating things. I can use my elbow to turn off the lights. And I'll just make my way back, just being careful not to disturb my layers. Okay, so I'll just make my way around here and spray it back in. Okay, so I set this carefully here. I'm going to spray in my new flask, which I've already labeled in advance. I'll set this over to the side. And now I want to get rid of my old medium. So actually, I need one more conical tube for this. So let me go ahead and grab that. This will just be a waste tube to dispose of my old contaminated medium in. I guess really I should label it. So now that I've sprayed it, I can't label it, but it's good just for demonstration what you can do if that happens. Just take a paper towel, dry it so that I don't ruin my little Sharpie. And I'm going to just label this waste. I can just put a W on the top in case the cap gets separated. Okay. So I'll bring that in. Now, if you have a larger conical tube and some smaller ones, you might have to just flip this and use them all on the larger side. Since this is just waste, it doesn't matter if I leave the lid off of it. I'm going to put it here. Okay, I'm going to take my conical tube, and hopefully you can still see I have my nice tropical smoothie layers, and right at the bottom, the little clump of cells. So I'm going to take off my lid, and I'm going to very carefully remove the old medium. Now, you could do this with a micropipette. All the cells are down at the bottom, so it doesn't matter if you remove the old medium with a micropipette, because the cells aren't going to be going through that. But I'm going to just use this serological pipette. I'm going to carefully withdraw the old medium, and you can, I try to keep the tip of the pipette near the top. I don't want to go all the way up to the top because remember there's a filter in here. If I draw the liquid all the way up to the top, I'll ruin my filter, which costs like 10 bucks. That's always annoying. So, and once again, I'm going to go down. I keep my tip near the surface of the liquid, and I just carefully withdraw a little more, a little more, a little more, until hopefully you don't really see any pink left. Okay. And now I should just see golden, maybe a tiny, tiny bit of pink at the top is not the end of the world. There we go. I've gotten rid of my DMSO containing medium, and now I'm ready to resuspend. So I'm going to set this down, set the cap back on, get my new medium, my brand new medium. Draw some up. Just set this here for the moment. And now to resuspend, I want to make sure that the pellet comes in and out of the tip of the serological pipette a few times. So I'll expel some medium, but I'm trying to make sure that that pellet of cells comes up. Sometimes it's a little stubborn and it doesn't want to come up. You can kind of gently blast away at it, and I saw it just kind of dissolve away, and now I'm sure my cells are pretty nicely mixed. Again, whenever you're working with cells, you don't want to draw it up and down at super speed, but at a moderate speed up and down is not going to hurt them. So I just gently mix. Once everything is all mixed together, I'm going to get my last little bit of medium from this other flask. I can do this quite quickly because it's just medium, no cells. Okay, that's everything out of there. All right, everything's all mixed together. And now I'm going to transfer it all into my new T25 flask. And just to make it a little bit easier, I could have done this a while ago, but I'm going to switch over and use a 10 ml flask instead of my 5 ml flask to make it easier. Once again, as I open it up, I try not to touch anything. I secure it firmly, set my trash over here to the side. I draw up my cells. I lost my cap way down there, but it's okay because I'm throwing that away anyways. I take my lid off. Once again, we try not to touch the tip to the rim here. Now you'll notice I just dropped a little drop. That's okay. I lost, a few million cells, but no big deal. I'll just clean it up at the end. I insert this here and gently expel everything inside. Okay. I put my lid on right away. Now, one thing that's important to note is the flask, the lid points slightly up. Please don't, be using the flask upside down. You can tell which side is top because the lid points slightly up and the rim around the top is thicker. Some lids are fancy and actually have a filter in the top. It's just kind of a white looking thing. These are less fancy. You don't want to screw it all the way shut. If you screw it all the way shut, then your cells can't breathe. You want to leave it a little bit loose such that you can gently pull it back and forth, but not so far that it's about to fall off. So you just kind of give it, enough of a turn that it's on there securely, but that air can still slowly make its way in and out so your cells can breathe. So I just gently put it back and forth to make sure that there's even coverage. And then I come over to the incubator. Now, when we put things in the incubator, I'm going to actually grab my ethanol. It's good practice to spray things as they go into the incubator to avoid, once again, contamination in the incubator. And there are actually several things I'm going to be looking for every time I open the incubator. One thing is I need to look at the water bath and make sure that the level is plenty high. Your cells will die if they dry out, and because everything is heated in here, it will dry out if you don't have plenty of water in that water bath. So make sure that the water bath is at least half full. If it's not, you can go refill it, again with the little special detergent that we add to prevent growth in there. If you look in and you see that there's kind of gross things swimming in the water down there, just go dump the whole thing down the drain, refill it, again with little, I'm blanking on what the name is, but the thing that you add to sterilize the water. So several things you want to be looking for. One is water. Two is always check the temperature. The temperature should be around 37 degrees, mammalian body temperature. Here it's 36 because I had the door open for a while, that's okay. And the carbon dioxide should be around 5%. Again, it's a little bit lower because I had it open, which is okay. It's adding it back. Where does the carbon dioxide come from? The carbon dioxide comes from this tank over here. There are two things to notice here. The left vial shows the pressure that's being allowed to go into the incubator. You'll actually notice that it's kind of jerks up and down. That's just because every time the incubator senses that it needs more carbon dioxide and releases a little, the pressure just drops when that happens and then it slowly goes back up. This needle on this side is measuring what's on the inside of the tank. You can see it's at a much higher pressure, about 900 psi, and that just shows how much is left in there. As it starts getting down, to the 200 level, just if you notice that, let me know, let Mr. Erickson know so we can make sure we have a new bottle of carbon dioxide ready to go. Okay, so if your humidity is good due to the water bath, it should be up at 100%. Just being exposed to that open water bath in the warm air. The temperature at 37 degrees Celsius, the carbon dioxide at 5%, you should be good to go. If you notice that the incubator is off, the power button is right here. If the power button is on but nothing is running, then that means we've probably tripped a circuit breaker. Let me know, let Mr. Erickson know right away so we can get the power restored so that we don't lose everything that's living in here.